Well, it's really my pleasure to introduce the speaker for this afternoon, Dr. Ben Sullivan. Ben just started at UNR last month, actually, and he was hired on after a very competitive search for a soil science position. We had over 120 candidates, I believe, and Ben rose quickly to the top of the list because of his incredible credentials. And he got his bachelor's in forest resource conservation at University of Montana in Missoula, and then got his master's and PhD at Northern Arizona University, and then did a postdoc back in Montana with Corey Cleveland. Ben has done a lot of very interesting work related to carbon and nitrogen cycling in soils and relating those, microbial processes to basic soil processes. And he's like I said, his resume is pretty amazing. He's published in journals such as Nature and Proceedings of National Academy of Sciences and ecology letters, which, not only in the field of ecology, but in any science field are really very high-profile journals. And so we're very pleased to have Ben join our department, and I think you'll be a fantastic addition to the faculty, both in terms of research and teaching. And so, without further ado, the floor is yours.
Well, yeah, thank you, Paul, and thank you, Laurel, as well, for the invitation to present here. This is a great opportunity to share some work that I've been doing over the last few years with y'all. I would like to start off with the blanket statement that soils are extremely important. They're really important, not just to me, but to all of us, because they're the growth medium for most of the food, fuel, and fiber that sustains our societies. Soils also perform a number of important ecosystem functions that maybe we can't even see, but they maintain our atmospheric, the growth of plant communities. Most of these ecosystem functions are mediated by bacteria, fungi, and even archaea in soil. And so I want to point out the tremendous biological diversity and biomass that occurs in soil. Within a single gram of soil, which is roughly the size of a quarter, it's a little bit lighter than a quarter dollar, you can have up to 10 million individual organisms in some extremely rich soils. That number can vary substantially between the different ecosystems that you might be in, and climates and things like that. But you get the idea. Soil is just teeming with life. The organisms in soil perform many functions. They're the reason, these organisms are the reason that we're not swimming in the detritus of our ancestors, because they decompose organic matter. They provide nitrogen to ecosystems via biological nitrogen fixation. And the decomposition of organic matter actually also releases nitrogen that's bound up in organic forms into inorganic forms that plants and microbes can use. Soils store a tremendous amount of carbon. Roughly 2.5 to 3 times the amount of carbon is stored in soils as occurs in all of our atmosphere at any given time. So soils and the microorganisms that live in them are extremely important to the global carbon cycle and the global nitrogen cycle, and also the hydrologic cycles. Of course, soil stores water and releases that water during periods when there's not been recent inputs. But even though arid and semi-arid ecosystems are home to roughly 40% of the Earth's human population and cover about 40% of the terrestrial land surface, we don't really, if we look at a global map of a biogeochemical process, we often see something that looks kind of like this. The arid and semi-arid regions of the Earth often are about the same, have the same color characteristic as ice-covered Greenland. Big white spots. It's easy to make out the Sonoran Desert, the Mojave Desert, Atacama Desert, Patagonian Desert, Sahara, you get the idea. We basically ignore deserts and arid ecosystems because it's easy to adhere to two principles. Nothing happens when it's dry in soils, and nobody's home. The microbial communities in arid soils and the processes they perform can easily be ignored when we're looking at big picture global rates. Now, I must point out that I stole this, or perhaps misappropriated this slide, from my colleagues and co-authors, Bill Smith and my postdoc advisor, Corey Cleveland, who generated this figure to show in an article in the Proceedings of the National Academy of Sciences how nitrogen and phosphorus support net primary productivity and may support primary productivity, plant growth in a future environment. And they were making the point that the tropics are really important, and they weren't trying to say that deserts are unimportant. So I've kind of cribbed this and used it for my own purposes. But the message remains the same. If we look at biogeochemical processes globally, they're often ignored. This may be a function of where we work. Now, when I say we, I don't necessarily mean us here in Nevada. We may be perhaps the bulwark against some of this. But if we look at this paper from Frontiers in the Ecology and the Environment a couple of years ago, very nicely demonstrated where ecologists work in the field relative to the expected or the land area of those areas. So they divided the land surface up into major biomes, and then in the blue bars measured the number of studies that occur in those areas. In the red bars, we see the expected land area, or what we'd expect, the number of studies we'd expect based on the relative land area of these different biomes. And so on the left side of this graph, we see that temperate evergreen woodlands and temperate deciduous woodlands are really well characterized relative to their land area. But when we move over here to the right side of the graph, we see that arid environments and semi-arid environments like savannas and shrublands and desert and barren areas are underrepresented in research relative to their land area, and in some cases, very significantly so. What I would suggest here that this kind of represents a humid ecosystem bias in our understanding of processes, and but not necessarily that we don't understand how arid ecosystems work. It's more just that what we know about humid ecosystems can overwhelm our understanding of arid ecosystems. And so there's sort of this perspective bias that may occur. That can be very nicely depicted in this box and arrow diagram of dry season and wet season biogeochemical processes. In this case, it's basically the nitrogen cycle. And we have very small arrows between boxes that verge into dashed arrows during the dry season. And then the rains come, and everything turns on, and everything gets active. All the microbes must get active, and this is what we're left with. This paradigm that in the dry season nothing happens, and it requires water, and that water is required for anything to occur. In some ways, it's very generous that they even use dashed arrows. They just needed to represent the arrows somehow in the dry season figure, because that's kind of how we consider the dry seasons here. When we think about arid ecosystems, we have to think about what available water actually might be. There's this sort of humid ecosystem bias to our perspective of soil processes in arid ecosystems. And so our concept of arid ecosystems as being water limited is kind of fundamental to how we view these ecosystems. We can go back 40 years to a definition of deserts as water-controlled ecosystems with infrequent, discrete, and largely unpredictable water inputs. Water-controlled ecosystems in the desert, and I'm not the only one who sort of is thinking about these kind of things. In a really eloquent piece a couple years ago, Amy Austin, who's also the author of that box and arrow model that I just showed a couple slides previously, asked if we basically, by focusing on water limitation in deserts and arid ecosystems, are kind of limiting ourselves to our understanding of the soil biogeochemical processes that occur. We have to ask ourselves what available water really actually is in a water-controlled ecosystem, and we have to consider the balance between the inputs and the outputs that occur in any environment when we start talking about available water. Of course, precipitation is going to be an important input, and then in an arid environment, evaporation and transpiration are also extremely important. So soil texture can really drive the outputs of water and the availability of precip to penetrate into the soil as an input. And so I show this image of black sand beach in Costa Rica, not because I want you to be jealous that I got to take that picture and stuff like that, but actually because this black sand beach like this is going to have very different infiltration capacity for precip, or in this case waves, than might a soil that's much more weathered and developed and has much finer soil texture. So when we consider the availability of water, we can consider the water content in the soil, which is presented here on the y-axis as the volume of water per volume of soil, or we can consider the amount of energy it takes to take up that water by a plant or a microbe, and we call that soil water potential, and that's on the x-axis. And this helps us understand what available water might be, because in a sand, you can in three different soil textures, presented here as sand to the lower left, an intermediately textured loam in the center line, and then a fine textured clay on the upper right line. We can have the exact same volume of water in the soil, in this case 20%, or 20%. And there can be very different energy required to take that water up. In a coarse textured sand, that energy required to take up the water might be low, and in a fine textured clay, there might take more energy to absorb that water. Another way to view this would be that from sort of and again, this is kind of a humid soils perspective, is that clays absorb a lot of water, but it might be harder to get, because you can have, at the same water potential on the x-axis, you can have 60% water in a clay, but maybe only 25% water in a sand. Or the other way to look at it is that a sand doesn't absorb as much water, but it's easier to get. So in the same paper that 40 years ago described deserts as water-controlled environments, Emmanuel Noirmire proposed the inverse texture hypothesis for water availability in arid ecosystems. And this suggests that the idea that clays are wetter than sands, which is how we view soil texture from a water holding capacity standpoint in a humid ecosystem, might not actually apply in an arid ecosystem where evaporation and transpiration outputs dominate. They're greater than the precipitation inputs. And so Noirmire asked the question, do sandy soils have more available water than a fine textured soil? And subsequent work suggested that ecosystems with about 370 millimeters of rain per year are kind of the threshold at which this inverse texture hypothesis would occur. The mechanism of the inverse texture hypothesis is basically that in an arid ecosystem, if a rain falls on a finely textured soil, it's going to evaporate before it can penetrate into the soil and be available to microbes and plants. Of course, in a fine textured soil, like that sandy beach that I showed, that water can penetrate down to depth where the plant roots are and the microbes are living, and can be utilized even though it might not. So basically the soil texture, the coarse texture, holds more water that's available to plants even though there might be less volume of water in the soil profile. So I worked on a really neat ecosystem in northern Arizona, a series of sites in northern Arizona, in Piñon juniper woodland ecosystem that gets about 336 millimeters of year, which is a little lower than the 370 millimeter threshold proposed for the occurrence of the inverse texture hypothesis. And I studied 4 different sites that had different ages since the soils began to form. Cinder cones erupted across a volcanic field, and the youngest site was 930 years young. The oldest site was 3 million years old. And during the course of soil development, soil gets more finely textured as it weathers. So I was able to explore how, working with an undergraduate student, he and I were able to explore how the soil texture may affect plant growth. And we were able to also take advantage of a 6-year watering experiment where we watered whole mature piñon trees and watched that response to water availability. So here's that gradient in soil texture that occurs over the course of 3 million years of soil development. We go from almost a complete sand at the earliest substrate age down to very little sand by 3 million years, and we've reached about 43% clay at 3 million years of soil development, which is a pretty substantial amount of clay in the soil. So we measured piñon growth on these 4 different sites with different soil textures, and also between watered and unwatered piñon trees. And so we looked at the growth of the trees from a number of different angles, but we hypothesized that if we got greater growth on sandy soils than clay soils, then we'd be observing the inverse texture hypothesis. Whereas if we had greater piñon response to watering in sandy soils, if we had greater piñon response to watering in sandy soils, then we'd also see an inverse texture hypothesis. The inverse texture hypothesis would be supported. So we'll begin by looking at the black bars here. I've taken away the substrate ages or the site ages on the bottom and just demonstrated them across the x-axis as sand to clay. And on the y-axis we have one metric of growth, which is leaf area production. And we can see, if we look at the black bars, the unwatered trees, that generally the more sandy side of the image has more growth than the finer textured side of the image. We get a similar effect when we look at branch elongation. So the last slide was leaves. This is branches. And when we go back and we look at the watering effect on the leaves, we can see that we basically get a big response in the sandiest site to watering. So these are kind of beginning to indicate to us that we may be seeing conditions consistent with the inverse texture hypothesis. We also got this really strange effect when we looked at piñon growth rings. And so after 6 years of watering these trees, we cored the trees and looked at the growth relative to the mean annual growth of the trees. And we found that the watered trees at one of the finer textured sites actually did worse than the unwatered trees, which, if you water a tree in an arid ecosystem, you'd expect it to grow more. And that's what happened at our sandy site. It grew a little bit more. The difference wasn't significant. But it was very interesting to see that the watering actually seemed to cause some kind of negative response in this one site, which kind of challenges this idea of water limitation in an arid ecosystem. We're not really sure of the mechanism that drove this, but it's challenging some of these paradigms that I've been talking about. So we saw some evidence that would really suggest that the inverse texture hypothesis occurs, and we were on the dry side of that 370 millimeters of precip per year threshold. So it makes sense that we would see the inverse texture hypothesis occurring here. It was nice to be able to show that across this gradient of soil texture and watering experiments. So when we think about what's plant available water in an arid ecosystem, we really have to consider the soil characteristics, and it may not work quite the same way as it does in an arid and a humid environment. So now I'm going to shift gears and talk about an important soil microbial process, decomposition, which, as I mentioned before, is the reason why we're not swimming in the waste of our ancestors. Decomposition removes the waste from any ecosystem and recycles nutrients. It also produces carbon dioxide naturally. It's always occurred. And the production of carbon dioxide from soils roughly balances with photosynthesis from plants and accounts for the carbon dioxide concentration in our atmosphere minus the addition that we've put in with fossil fuels. We can, in most ecosystems, really nicely summarized in this science paper from a few years ago by Bill Parton and colleagues, show that water and temperature are really dominant controls on decomposition processes. And I think that would make sense to most of you. If you heat something up, the microbial activity is going to increase. And if you add water, there's going to be more activity as well, less dehydration of the cells. And so you get more decomposition. It's actually really easy to model, well, relatively easy to model decomposition compared to some ecosystem processes. And so, but one notable holdout from this paper was arid and semi-arid environments that just didn't follow along with the model presented here based on water and temperature. And so these figures on the right come from the paper that I've already cited by Amy Austin in 2011. And it demonstrates quite nicely that decomposition in arid ecosystems outpaces what we would expect based on these models developed from humid ecosystems. Let me make sure that this still works. Excellent. So let's focus on the top figure here. And we can see that basically regardless of the annual precipitation, which is presented on the x-axis, we have a baseline level of decomposition that occurs. And in fact, somewhere around that 336 millimeter threshold of precip that I worked at, where I saw the inverse texture hypothesis, you can actually go drier and find higher rates of decomposition that occurs. And if we replot this as the expected decomposition based upon models on the bottom here, on the x-axis, in the bottom figure, and the observed decomposition on the y-axis, we see that the observed decomposition is greater than the expected decomposition, and it's above this 1-to-1 line. And the drier that you get from 0 to 100 millimeters, the farther left you are from the 1-to-1 line. And the wetter that you get from 250 millimeters of precip per year up to 500 millimeters, the closer you get to the 1-to-1 line. So why is this, that these arid ecosystems are actually having much more decomposition that occurs than we would expect? Well, the answer is kind of a strange one, but it's also an intuitive one, which is often how these things work. I'm going backwards. There we go. The reason that this occurs is photodegradation. So the sun's rays essentially oxidize the organic matter just the way, in a photochemical way, rather than a biological way. And so if we think about an arid ecosystem pictured here on the left and a humid ecosystem, perhaps a northeastern hardwood forest that's so well studied on the right, we can think about this as though when it's wet, biotic decomposition dominates in the humid forest. When it's wet in an arid environment, biotic decomposition is also really important. But typically it's not wet, it's sunny, and it's dry. And so in these ecosystems, you can have lots of photooxidation that's occurring in periods when we wouldn't expect any decomposition to be happening. So the combination of photodegradation and biological decomposition combine to increase decomposition much higher than we might expect otherwise. So decomposition in arid ecosystems actually outpaces some of the wetter ecosystems because they can take advantage, or they're essentially subject to both photooxidation and biological decomposition. Now here's another ecosystem process that's a really important one, and often you don't hear very much about it, perhaps less than decomposition, unless you spend a lot of time thinking about the nitrogen cycle, which, being that it's 4:30 on a Friday afternoon, I'm not going to take you through the nuances of it, but I want to point out nitrification down here on the bottom. It's kind of the middle link of the nitrogen cycle, and it converts one form of inorganic ammonium, that's inorganic nitrogen, ammonium, that's available to plants and microorganisms, into another form of inorganic nitrogen that's also available to plants and organisms, and that's nitrates, which is up here. So nitrification is kind of a two-step process, and I've circled the first one here for simplicity. So it's the conversion, as I've shown here, from ammonium to nitrate. Nitrate is kind of the last stop at which nitrogen is available for plant uptake. And we see that in most ecosystems, nitrogen limits plant growth. Plants need the nitrogen, need nitrogen to grow, and they have to get it from typically these inorganic sources. So nitrate can either be leached from the soils, or it can be lost as converted to a gas and lost from the soil. And so that's why I've called it here the last stop for plant uptake. And so nitrification is a pretty important process in regulating the amount of nitrogen in the system. So it's important to understand the controls of nitrification in soils. And one dogma of nitrification is that you have to have substrate, that's ammonium, makes sense, and you have to have water, because the nitrifiers, the microorganisms that are responsible for nitrification, are, well, they can't dry out, right? They shouldn't be desiccated and dead. They wouldn't be nitrifying. So you need some amount of water, and you need some amount of substrate, and the water is also sort of a medium in which the substrate is transported around in the soil. So these two things combine to control nitrification rates. And this figure on the lower right has water potential along the x-axis. That's the energy required to take up the water in the soil. And it has the fraction of, well, it's the fraction of the decline in nitrification rates on the y-axis. But if we look at roughly a 50% decline in nitrification rate starts to occur in pretty wet soil. We're still pretty far along, so the zero on this y-axis, or x-axis, excuse me, is off to the right, which is kind of funny. Negative, water potentials are negative, of course. And so we're still dealing with fairly wet soil here, and yet we've declined nitrification by half. And so water limitation becomes a really important component of nitrification controls. This was work done in California grasslands, which were more humid than many of the ecosystems that we deal with here in Nevada. And they're more humid than the ecosystems that I worked on in northern Arizona as well. So as soils develop, they have a soil textural gradient. They also have a soil nitrogen gradient that can occur. And I should point out that these sites experience a strong monsoonal seasonal dynamic pattern. So for part of this growing season, it's quite dry, and then the monsoon season rolls in and we get lots of precipitation and wets up the ecosystem. So we get a kind of bimodal precipitation pattern where we have dry times of the year and wet times of the year. So this is that soil nitrogen gradient that I mentioned. We get an increase from very young, increase in nitrogen, total soil nitrogen from very young soils to 750,000 years. We get a decrease to 3 million years. There's a whole talk I could give on just why that occurs. So I won't go into the details, but we see this sort of gradient of soil nitrogen across this soil chronosequence, it's called. So I decided to study the controls of nitrification along this gradient of substrate supply, because we have this gradient in nutrient availability, and in two different time periods, the dry season and the wet season. And to do this, I decided to use nitrification potentials, which are a really elegant technique that basically provides abundant ammonium. So you're just swamping the nitrifiers with all the food they could want. You then put this soil, whether it was dry or wet when you collected it, you put it in water and you shake it up, and that provides the water for the microbes to use. It also allows the ammonium to move around in the solution. And because nitrifiers are so generally slow growing relative to many microbial communities, what we see, the rate of nitrification that we see in this assay should generally represent the potential of the microbial community to have nitrified soil based on what we collected in the field. So it's kind of a maximum capacity, because we've given them lots of ammonium, for nitrification in the soil in those field conditions. And so it might also be an indicator of the microbial community size. So what we should see in this assay is very little nitrification in the dry season and much more nitrification in the wet season. What I actually measured was a lot of nitrification in the dry season, especially at these two middle sites, and less nitrification in the wet season. This is very strange. The other two sites were essentially the same between the dry season and the wet season. So I started wondering why this occurs. And while I was considering this and even starting to write the manuscript, two other folks began to consider the same idea, because they had found the same pattern. And this came out roughly the month that I was ready to start submitting my manuscript. And so sort of caused a redirection in writing the paper. But basically, Parker and Schimmel demonstrated that they found higher levels of nitrogen in soil in the dry season than the wet season. And critically, just like I did, they measured greater rates of nitrification potential in the dry season than the wet seasons. Okay? So now I know, well, at least what I measured wasn't completely crazy. But they also provided a really elegant mechanism that explained why they think this might occur. And so on the upper figure here, they have a wet season, several soil particles in gray, in the wet season, connected by water. And they have ammonium in there. It's probably impossible to read, but there's ammonium in there, and there's some nitrifiers in there, but there are also predators, and they call them agents of death. And they say that in the wet season, the nitrifying bacteria are not isolated from these agents of death. So basically, predation is occurring and knocking down the nitrification community in the wet season. But in the dry season, the nitrifiers can hang out on these very fine films of soil water, water on the soil particle, where there's ammonium, and yet they are sort of protected, physically protected from predation. Well, that seems kind of reasonable, but I also sort of made me think, well, sure, maybe there's tons of potential for nitrification, but maybe that just means that the microbial community is somehow subsisting through the dry season and not very active. And so I proposed that basically what we see is sort of an empty pipe in which there's the potential for lots of things to occur once the water does come, but basically not a lot might be going on. Whereas in the wet season, there might be less going on or less potential because the microbial community is getting predated upon or something like that. But there actually could be some rates occurring. So these are full pipes. They just happen to be smaller arrows than the empty pipes that I showed on the last slide. So it's not really clear if dry soils have more active or more nitrification that actually occurs in the field, and so that's something that we need to continue to follow up on. Another alternative or another mechanism that could potentially explain this process is that nitrifying bacteria aren't the only ones out there playing the game. So has anybody here ever heard of Craig Venter? No? Okay. Craig Venter was basically a big part of the drive behind sequencing the human genome. And so he developed a lot of techniques to be able to sequence a lot of DNA really quickly. And he went all over the world and started sequencing things. And somewhere out in the Sargasso Sea, he sequenced some water and found some archaea that had the exact same sequence as nitrifying bacteria. It basically encodes for the gene that creates the nitrifying enzyme. And so suddenly, about now probably 10 years ago, suddenly archaea burst onto the scene as a potential mechanism for nitrification. And so actually at the same site that I worked at, one of the sites that I worked at, the youngest site that I worked at, Karen Adair from Northern Arizona University went out and sequenced or measured the amount of sequences, genetic sequences, presented here on the x-axis on a log scale, of bacteria and archaea that are capable of nitrifying in both the dry season and the wet season. And she found that archaea are always more dominant in either season than bacteria, so they're a key part of the nitrification process. And she did see some pretty interesting and significant differences in which there were more archaea and less bacteria in the dry season. So there could be kind of a microbial community compositional effect here as well. It's also interesting to consider that this may not actually be exclusively limited to arid ecosystems. So I got to do my postdoc in Costa Rica, studying how nitrogen and phosphorus affect tropical forests. But I worked with folks who studied the nitrate concentrations in rivers. And they found something really interesting. So when they measured very fine-scale resolution, temporal resolution, the nitrate concentration in a Costa Rican river, they found that during the wettest time of the year, nitrate concentrations were lowest. And in the dry season, which is actually several months long in some tropical forests and in the one that we worked in here, we got the highest concentrations of nitrate. And the mechanism that they invoked to explain this was that basically nitrification continues during the entire dry season, as we saw in northern Arizona semi-arid ecosystems, but that once some kind of pulse precipitation event occurs in the dry season or towards the end of the dry season it starts to rain again, you get hydrologic conductivity that's essentially flushing this nitrate that's been developed in the soils down into the rivers where they can measure it. And so this nitrification may persist in the dry seasons in these rainforests, even though it gets very dry, and most people would, like in arid ecosystems, think that not much is actually going on. The forests where this occurred receive roughly 3.5 meters of precipitation a year, which is, 35 centimeters, an order of magnitude more precip than than were the sites I worked on in northern Arizona. So nitrifiers may actually prefer dry conditions because they have less competition for ammonium with plant uptake and other microbes and stuff like that. They may not be eaten by agents of death, which is always good if you're a nitrifier, I guess, and or anybody. Maybe, and what this pattern that we observe in arid soils may actually be general, as evidenced by the Costa Rican River data. And it may be potentially mediated by archaea, which we've just begun to understand. So I'm going to very quickly take you through one last ecosystem process that I'm very interested in, which is the consumption of methane by microorganisms in soil. Methane is important because the concentration in the atmosphere has risen sharply since pre-industrial times. Methane often gets associated with livestock production, but though there are lots of ways in which it ends up in our atmosphere from anthropogenic activities. But it also, even though it occurs in less concentrations in the atmosphere than carbon dioxide, which is kind of the famous greenhouse gas, it has 25 to 30 times, depending on who you talk to, the heat trapping potential of carbon dioxide. So methane's kind of, it's been called the second most important greenhouse gas behind carbon dioxide, if you ignore water vapor, which we shouldn't. But this concept is that methane is consumed by microorganisms in the soil, and this methane diffuses from the atmosphere into the soil profile. And this process has been widely believed for decades to be driven by the quantity of air-filled soil pore space, which is a function of soil texture and soil water content, because if you have coarse texture, you might have lots of big pores, and if you have fine texture, you might have lots of smaller pores. And water fills in those pores and becomes water-filled pore space. But here we're talking about gas diffusing in from the atmosphere, so you need that air-filled pore space. You also need oxygen because it's an aerobic process, and of course you need the substrate from the atmosphere, the methane. So these are the kind of three controls that people invoke for methane uptake in soils. So just keep in mind, I always kind of imagine like a little Pac-Man guy munching methane in the soil. What has been actually modeled, I sort of took this from a paper by Steve Del Grosso and colleagues. This is a general pattern they proposed and used in a global model of methane uptake in soils that essentially at some low level of soil water content, there's cell desiccation. So these Pac-Man are drying up and not able to consume methane. But then after they have enough water, but not too much water to fill the soil pore space, there are optimum conditions for methane uptake. And then as you continue to fill the soil pore space with water, methane uptake decreases. So that concept of dehydration or desiccation of the methanotrophs, the microorganisms that eat methane, was bolstered in arid ecosystems. This was in southern Nevada, outside of Las Vegas, by a paper back in 1992 that was published in Nature that demonstrated quite elegantly that whenever it was dry, you had low rates of methane uptake, but as soon as you added a little bit of water through a precipitation event, methane uptake increased. So that would typically be evidence for desiccation. So I decided to test this same process along this age gradient that I worked on for my PhD that had these strong wet and dry seasons. And I used potentials just like for nitrification, but in this case, it was methane uptake potentials. I'm going to move quickly through this, but essentially it's the same idea as the nitrification potential. It's a measure of the community size, and it's like the total maximum capacity of what's in the site to oxidize or consume methane. So here's that strong soil texture gradient again, when texture is an important component of air-filled pore space. And what I observed was that in the wet season, we got more methane uptake than in the dry season, which would be consistent with dehydration or desiccation of the methanotroph community. Except I want to point out that in the dry season, we had the most methane uptake at the coarse textured site, where we actually had less than 1% water content. So remember I said that sandy soils don't hold quite as much of a volume of water? Well, here they don't hold very much water at all. This is an amazingly low amount of water. So somehow, there's a lot of methane uptake in the dry season in extremely dry soil conditions. That is not consistent with desiccation. So something weird is going on here. And again, I kind of scratched my head and spent a while trying to write the paper and figure out the story. And I was continuing to do some experiments and look at the data. And so I went back and I focused more closely on relationships between water content and methane oxidation. So this is gravimetric water content. It's the mass of soil, or mass of water per mass of soil. And we have the dry season in the open circles in the dashed line and the wet season in the full circles. And we see that we see two opposing patterns. Again, not consistent with desiccation. So desiccation or dehydration of the methanotroph community may not be going on, but in the process of analyzing my data, I went back and considered the dissolved organic carbon concentrations in the soil that I had sampled. And I found it, well, for a soil biogeochemist perspective, a fairly strong R-square, maybe not for an engineer, but an R-square of roughly 0.6, suggesting that 60% of the variation in methane uptake was driven by dissolved organic carbon concentration. Now this is really weird because methanotrophs have long been believed to be obligate methanotrophs. That's all they do. They just munch methane like Pac-Man. Why are they related to dissolved organic carbon concentration? Well, looking into the literature and some further analyses really indicated that methanotrophs may be facultative. That is, they may be using dissolved organic carbon during some conditions and then also consuming methane in other conditions. I've recently begun working with a research scientist at the University of New Hampshire who added leachate, leaf litter leachate, to stream water and stream sediment. And these two figures, at 60 minutes and at 120 minutes on the right, divided by this dashed line in the middle. On the x-axis, we have all the different leachates, and we have methane concentration of the atmosphere in a jar, basically. On the y-axis, these two figures might look similar at first blush, but what we see is that with some leachates, especially Russian olive and oak and I think cottonwood, we get a decline in the methane concentration in the headspace, indicating methane uptake as a result of leachate addition. Notably, the controls stay really similar. So this is an indicator that even in riparian ecosystems, this may actually be continuing, that this relationship between dissolved organic carbon and methane uptake may be important. So methanotrophs may prefer wetter conditions, but it may not be for the reasons that we thought. And I hope that by showing you all these different soil processes and microbially mediated biogeochemical processes, I have convinced you that basically the white spaces that we see on a global map may not be appropriate. There are processes occurring in there that are really important.